great. Hi, my name is Emily. I am a librarian at AADL, but one of the best parts of what I do is I get to be here and MC Nerd Nite, and we have got a great night planned for us tonight. So we'll begin, like I said, AADL. I also want to say a huge thanks to Live. What a cool thing that we get to do every month, that this nightclub opens early for us so that we can come and learn but also have a cocktail. It's perfect. So thank you to Live, and make sure you tip your bartenders well, because they are making it so that we can be here. And of course, we wouldn't be here if it wasn't for you all. So thank you for coming out despite the construction in very inconvenient places, despite the fact that it's cold and the world is cold and sometimes it's hard to get out and do something. But y'all did something tonight, so thank you for coming. But we also would not have Nerd Nite if it wasn't for our wonderful speakers. Tonight we have Dr. Ajay Larkin talking about critical hits in medicine, how to find the weak points of a multidrug resistant fungal pathogen. We have Justin Shell with Reckoning of Deeds of Injustice. This is a sequel to a talk that I think was 3 years ago, so we all get to learn a little more. And Ananya Sen talking about how oxygen is good for you. Or is it? But first, it's time for your Nerd Nite Minute. Did you all know that we had a zoo? Ann Arbor had a zoo. But I do want to give you this disclaimer. The zoo was open in the 1930s to 1960s, opened in 1929, which is maybe not the best time for zoos, and is kind of a bummer, especially when you look at it through today's sensibilities. So I want to acknowledge that up front. But now, set that a little bit aside and introduce you to some of the folks who were around Ann Arbor Zoo. So this is the zoo. This photo is actually from 1960, towards the end of its time. It was on Washtenaw and Forest, kind of like where the big student bus depot is right now. And it was the University Zoo. But it was also built there in theory because it would be able to spread cheer to the children who were in the hospital. But it spread cheer to children who weren't in the hospital as well, as they gathered to peer through the fences and see who there was to see. This is Trappy. Trappy is a wolverine. Trappy's name actually was Intrepid, but they very quickly realized it was a terrible name for Trappy because Trappy was very, very shy. And so maybe didn't live up to their name, but this is a photo of Trappy from 1946. They also had otters. This is an otter who came to join the zoo in 1958. The newspapers talked about how it had its own pool and slide, and people were really excited to see the otter. You know who else was really excited to see the otter? The raccoons who lived next door. There they are. How come they get a pool and we're just here? But the real draw, the real draw that you can read about in the archives now, the most out of anyone else, are these bear cubs who joined the zoo in 1957. And even in the 1950s, when you have something that needs a name, why, of course you have a contest for school children. But they were very, very strict in their announcement. They did not want you to call in and give them names. You had to send a postcard, and they spent quite a while talking about how you really did have to send in a postcard. But good news, this kid sent in a postcard. Little William there, he was 13, and he named them Maize and Blue. So he was maybe stacking the deck a little bit in this town. They ended up calling Maize Maisie. Maisie was the girl, Blue was the boy, and they lived happily in the zoo. And then comes 1962, and the university, as the universities want to do, wanted that land to build more of a building. And so they needed to close the zoo. And so they announced that they were closing the zoo and were looking for homes for these animals. And the leader of it got all sorts of interest. They said he got more than 200 letters and approximately 50 phone calls. Only one letter came from people in Ann Arbor, but there were many, many Ann Arbor phone calls. But my favorite letter of the ones that they put in was from Betsy Darty of Huntington, West Virginia, who said, "Dear Mr. Irving G. Reitman, how are you? I am good. I saw in Mama's paper about you were giving away animals. Would you please send me a skunk? Mama says it's okay. Thank you." Believe it or not, they did not send Betsy a skunk. They ended up deciding that they were going to send all the animals to a zoo in Omaha. And they said that they picked this one because it looked to have the best treatment in place for the animals, and that people would also still be able to see them for free, just like they were in Ann Arbor. Now, working with an archive, because I was not alive in 1962, something happened, and instead of every animal going to Omaha, Maisie and Blue, our bears, were getting new homes I got to look at my notes here, in Athens, Michigan. And so this is a picture from the first day that they tried to get Maisie and Blue to go to Athens. But these bears did not want to go into the bear crate. I can't blame them. It was not a huge crate. They did try using a prodding stick, but the stick did not work, and so the next day they tried the carrot, and the carrot being food. They started out using water-soaked raisins, and believe it or not, the wet raisins didn't work, despite the fact that they said they were a favorite snack. But eventually, warmed maple syrup drew them in, and so they were on their way to their new homes. So then there's the zoo. This is where they're starting construction for the university building, zoo building in the background. But I have one little epilogue, which is about our friends Maisie and Blue, who, when they went to Athens, guess what? They got renamed in a contest for children. And their names were Athena and Dokamak. And they had a baby. And I can only hope the very end of the article, which print is way too small to see here, mentions that Athens was the state's largest producer of maple syrup. So I can only hope that they heated some up for them as a present to new parenthood. And that is the story, very briefly, of Ann Arbor Zoo and your Nerd Nite Minute. So please continue that applause. Help me to welcome Dr. Ajay Larkin. They are part of Michigan Medicine Department of Microbiology and Immunology, where he studies gene essentiality, gene regulation, and high-frequency promiscuous gene incorporation in the emerging fungal pathogen Candida auris. Whew. Good stuff. Dr. Larkin received his Ph.D. in molecular and cell biology from Brandeis University in Waltham, Massachusetts. Join me in welcoming Ajay Larkin.
Thank you. All right, let's test this out. All right, thank you, Emily, for that introduction. I guess I didn't need an intro slide. Hi, everyone. Welcome to the first talk of the night. I hope you all have fun. Today I'm going to present critical hits in medicine, how to find the weak points of a multi-drug resistant fungal pathogen. It is indeed a mouthful, but that's just because I have a lot I want to share. So real quick up the top, I can skip a lot of this, but I work as a postdoc in Teresa O'Mara's lab down at the hospital. I do have a PhD in molecular and cell biology, and I have over 8 years of experience in my field. What I love is fungi and epigenetics, and today I'm going to focus on the first one. So why am I interested in fungi? Well, some fungi make us sick. So what happens when you get an infection? You either get bacteria in your body or fungi, and real quick in your head, if your doctor tells you you have an infection, how do you feel? Probably not panicked. You just think, ew, gross, I'll take medicine. And most of the time, you're right. We have antibiotics for bacteria. We have antifungals for fungi. Great. But even though we have this medicine, how often do you think about how this medicine works? And let me take it back one step further. How do we develop this medicine? How do we make it? So if we want to make something that attacks a pathogen, like, say, a fungi, because I like fungi, what makes a good antifungal? Well, every cell has genes, and these genes produce proteins, like this little squiggly ribbon thing down here. That's CYP51A1. It helps make the cell wall. That's the thing around the fungus that just keeps out all the bad stuff, keeps in all the good stuff. So these genes make proteins that help the cell survive. Antifungals work to prevent those proteins from functioning by blocking their function in some cool chemical or biological way. So antifungals block protein function, but what makes a really good antifungal is if it meets several different criteria. One is that it's target specific. Sometimes a fungal protein looks like a human protein, and if they match too much, then the drug will make you sick as well. So we want to find something that is specific to the fungi. Not only that, we want a high clearance rate. This is the critical hit. We want to stop proteins that the fungus absolutely needs to survive. These are important genes. So then the question you should all be asking is, well, how do we know if a gene's important? So let's think about ourselves for a second. We're human beings. We have a lot of organs. Some of them are essential. You can't really live without your heart. You can't live without your lungs. Most people can't live without their brain, but some of us get along okay. In the same way, fungi have essential genes. And a lot of these, I think about as a biologist, they're things that actually make sense for survival. Some essential genes build the cell wall. I mean, you couldn't probably live without your skin. Fungus feels the same way. There are genes that have to do with maintaining DNA. Super important. We've all seen DNA mutate in Marvel movies. And cell replication. If cells can't replicate, they stagnate and die. So, or disclaimer up top is, just because a gene is essential in one fungus, it doesn't necessarily mean that gene is essential in another fungus. So we can't just study one fungus and cover them all. So why are we interested in making antifungals? Well, they make us sick, and they cause over 1.5 million deaths a year. And that's bad. Candida auris specifically is a fungus that I study, and you can see at the top of the leaderboard, as determined by the CDC, it's one of the critical pathogens that they want to know more about. It's because Candida auris causes infections in humans that are already sick. It's the fourth most common nosocomial bloodstream infection in the US. That means that it's the fourth most common infection that you can get in a hospital, nosocomial, and once it goes systemic, it's associated with 30 to 40% mortality. So a serious problem, bringing down the mood a bit. So why am I talking about Candida auris? What's the deal with Candida auris? Is super sticky. It doesn't just stick to living things, but it also sticks to abiotic surfaces like hospital equipment, so it spreads a lot. It's multidrug resistant. And current antifungals that we use, they aren't super effective against Candida auris. So if you get it, it's a little harder to treat. And it also has an uncharacterized genome. We don't really know which genes are essential because we haven't really known it existed except for the past less than 20 years. And again, here's that disclaimer. We don't know what genes are essential in Candida auris because genes essential in other fungi aren't necessarily essential in this fungi. So we need to learn more about Candida auris. So what gene do we target to make a great antifungal? If Candida auris had two genes, like right here, you just point and say, the green one, essential gene, get that one. So if you're like me and you want to take the easy route, you'd think, can we just guess, just make a drug, let's find out if it works. But the problem is drug development takes years and a lot of funding. And what are the odds of finding the wrong gene in Candida auris? Well, Candida auris has about a little over 5,500 genes inside of it. And to make matters worse, we don't even know our odds because we don't know how many of these are essential. So that leads us to my project question. How do we know which genes are essential in Candida auris? And to tell you about this, I'm going to make a very logical transition. I'm going to talk about World War II. So in World War II, the US wanted to reinforce their planes. And if you're on Twitter, you probably have seen this a million times. Bear with me. So they want to reinforce their planes. However, armor is heavy and planes need to be light. So they wanted to prioritize protecting the essential parts of the plane. So what engineers did is they recorded where returning planes had been shot during battle. And based on this pattern of bullet holes, in your head, just think, where are the essential parts of the plane that they should protect? Thank you from the audience. Absolutely, places that haven't been shot. And that's exactly right. That's what you want to protect. And if that isn't clear, I hope to clear it up with a little example of how they took this data. So let's just look at this. You have a plane, it's out in the battlefield. There it goes. Somebody in the infantry fires an entire bullet at the plane. And it goes back to the hangar, and an engineer says, got shot in the wing. Perfect. Sure, that makes sense. What if a plane gets shot in an essential area? Well, it goes out, la-di-da. Oops, you hit my tail. You never make it back home, so there's nothing to record. So these blank spots in the data help mark the essential regions of the plane. And these blank spots in data can be really useful for any kind of essentiality mapping. So let's use this to learn about Candida auris. All we need is a molecular level machine gun. And luckily, we have that. So Candida auris has a bunch of genes, and we have a technique in lab called random insertion transgenesis. With this, we can take a piece of DNA that we call a transgene in orange. The purpose of this gene is that we can give it to Candida auris, and it will place it into its genome, breaking one of its own genes. That's the bullet. What we can do in the lab is we can treat our Candida auris with a little bit of electricity to do something called electroporation. It breaks open the cell just enough for our transgene to sneak in. Once it's inside, it acts as a bullet and is incorporated somewhere into the Candida auris genome. That disrupts a gene sometimes and knocks it out. And instead, now we have this transgene, which is the theoretical molecular bullet hole. So how do we figure out where we've shot Candida auris? Well, I'll go back to the exact same example. We're just using a DNA sequencer instead of a plane hanger. We have our Candida auris cell. Come on, buddy, get shot. Okay, never mind. That one made it. He's fine. We'll pretend it got shot because we're all friends. We place it into the DNA sequencer, and we take a look at it. The DNA sequencer says, hey, guess what? We shot it here. Okay, great. That's one bullet hole. Let's do this on a lot of different cells and do this experiment in bulk. And let's collect as many representative bullet holes, there you go, as possible. Place it in the DNA sequencer. There they go. And then up, they're recording all the bullet holes now. Perfect. But now, let's go back into it. What happens if we accidentally, or on purpose in this case, shoot an essential gene? Well, it never makes it. So it's never recorded, and again, we have that blank space. And so by doing this, we can map out all the blank spaces from our technique and find candidate essential genes. So to recap, we have a lot of genes in Candida auris, but we have a molecular minigun that we can use to just shoot out genes until we have these blank spaces. The genes that we shoot out that the cells can survive with are non-essential, and the blank spots are great candidates for future antifungal development, which hopefully, over time, we can make antifungals that can attack Candida auris' weak point for massive damage. All right, and I'm happy to take any questions. Thank you for your attention. Sorry? You got it. All right, yes, you. That's a great question, because what we call that, if you delete multiple genes and they die, that's synthetic lethality. That, with our current method, is not something that we look for. They fall out, and we never get to see that. So that requires a lot more experimentation. But that is an excellent question, and definitely something that we're always interested in. Yes, in the pink. Yes, I'll repeat the question. So the question I got was, what is a fungus in comparison to a bacteria or a virus? Well, there's a couple things. A virus actually is very minimal and can't self-replicate. So it's kind of like the minimal kind of thing that you need in order to deliver DNA or RNA to something to replicate. A bacteria is a prokaryote, which means that it doesn't have membranes for its organelles, little cell organs inside of the cell body. And a fungus has a lot of fungal-specific genes, but it is a eukaryote. It has bounded organelles and looks a lot more like other eukaryotic cells, including the ones that make up humans. Yes, over here in the corner. That's a great question. So the question was how, and correct me if I missed something. So the question was how one size fits all is this study? So for a gene, if I understand, that we find in Candida auris, how broadly applicable is it to other pathogenic fungi, maybe? Is that right? A different kind of fungus. Ah. Mm. Sure, that's also a great question. So the question, if I understand it more specifically, is about human hosts. So if you develop a drug and they have different, and those humans have differences, then maybe that might get a little dicey if you have one human that you didn't account for. And that's a good question. My field isn't as directly in drug discovery, but there are rare cases where you might have a variant of a gene, which is entirely possible. What we do, and one of the things that we do in the study, is we try and take a host of human proteins that have been studied and cataloged, and we can look at a database of human protein structures and compare the genes that we get from Candida auris to those structures to see if there's any similarity. And at least on our side, that's as much as we can do. But for the people designing the drugs, they might have something else that I'm not as familiar with. But it's a great question. Thank you. Yes, sorry. I don't know who was first. I'll just count on the audio. Yep, I can do both then. That is an excellent question because the typical answer isn't what applies in this case. So the question is, what allows the transgene to get into the genome? Now, typically, what we use with this method is called the transposon. It's very similar to viruses. It's something that allows for copy and pasting or cut and pasting of a DNA sequence into the genome. Now, that's a pretty common technique, but Candida auris is a little special and it doesn't quite express and read things the same way as a lot of other things we study. We found a very weird trait in Candida auris where if we just give it the transgene, it treats it as if it's a transposon as far as we can tell. That the gene just gets sucked up and placed somewhere randomly. If you play video games, I like describing it as Kirby storing it away for later. But as of now, how that works is actually another project that I'm just, actually in the process of starting. So it's an excellent question. Thank you. And yes. So the question is this my first rodeo with studying essential genes and designing a drug? And I'll tell you, this is my first time, and I am just a cog in the machine. I'm not a one-man pharmacologist. I would love to design a drug someday, and we'll see what happens in the future. But this is my first time using a method like this. Mm. So as of now, this is very early, but it is an excellent question. But it is a multi-step process. We'll probably have many people working on this for many years to come. All right, last question. Mm-hmm. So the question is, what do I think about corporations, groups that are designing surfaces to combat this? Actually, my boss shared, I think yesterday, a paper that showed a liquid-treated catheter that could decrease Candida auris's adherence inside of it, which is super cool. And in the time I spent to skim the paper, sorry, Teresa, and that it seemed like it also helped with clearing the infection because there was no place for Candida auris to set up a backup population. So treating surfaces, absolutely a very cool approach. Thank you. Great question. All right, thanks everyone.
All right, folks, we're going to take a 10-minute break, so I'll see you back here at 7:36. Go refresh your drinks, talk to your neighbors, and we'll see you soon.
Okay, that seems better. Hi, folks. Hope you enjoyed the break. Hope you guys got a chance to refill your drinks. We love our staff here at Live. We always appreciate them for hosting us. Give them a round of applause. And please feel free to stop back there and get yourself yet another refill. And, of course, make sure you tip our friends back there. Moving on to our second speaker, Justin Schell is going to give us an update of the Justice Indeed project. We just talked about targeting and microbes and things like that. We're going to go to a different form of targeting, and this is the bad one. This is racially restrictive covenants in Washtenaw County, which is something that we do need to address. There are way too many of them. So without further ado, Justin is going to give us an update. And are you going to share your cats? Are you going to talk about your cats? Please talk about your cats. We love your cats. And without further ado, Justin Schell.
So I will just talk about my cats. I do not have pictures of them, but find me afterwards, and I will show you pictures of my cats, Scanner and Pixel. They're both calicos, which means they're the riot girls of cats, as my friend says, called them. So thanks everybody for coming tonight. Thank you for Nerd Nite for having me back. I did talk about this back in 2000. I almost said 2003, 2023. Was anybody at that Nerd Nite talk? It was over at Venue. Yeah, awesome. Hey, awesome. So, what I'm going to talk about tonight, these 5 things. So I'll give a little bit of background, sort of refresh about the Justice Indeed project. We're going to talk about what racist covenants are, and just a heads up, you'll see some racist language. So take care of yourself if you need to. It's going to go by relatively quickly, but it's still there. And also, if you want to scan that QR code, that goes to the slides, which have a bunch of links on them to the different tools and things that we've published at the end, but I'll also have that QR code up at the end as well. I'll talk about some project results, because back in 2023, we were like, we're going to have this crowdsourcing project where you can help us transcribe all of these things. And then that happened, and then we had a whole bunch of people help us out. So I'm going to share some results from that, and talk about some publications and tools that are helping our communities reckon with these covenants and the consequences that they've wrought almost 100 years after some of them were put in place. And finally, we'll talk a little bit about what's next. So Justice Indeed is a community university project. It's some folks in the law school, in the library like myself, the architecture urban planning school, as well as our partners Future Root, that is Jessica A.S. Letaw and Yodit Mesvin-Johnson. I should say Mike Steinberg from the law school, Rob Goodspeed from architecture, urban design, urban planning, an advisory board, and lots of community members that helped us out with this project. So these are racist covenants. These are clauses in deeds, housing deeds. Usually they go back to the late 19th century, that were in a long list of things that said that what you couldn't do with your property. Sometimes this was no fraternities. Sometimes this was no liquor being sold. Sometimes this was you could only have this much space between the house and the street. But they also wanted to have racism in there. And so you see some of these things. These lots shall not be used nor sold to people not of the Caucasian race. A lot of white folks couldn't spell. No persons or any race of any race other than the Caucasian race shall use or occupy any building in any lot, except that this covenant shall not prevent occupancy by domestic servants of a different race domiciled with an owner or tenant. So you could be not white. Primarily, you could be black and live in this space if you were a servant. We see these all around the country. And this is a great documentary of a project in Minnesota called Jim Crow of the North, and talks about how these practices were more subtle, but just as nefarious in creating the segregation that we see today. We will see things, said premises shall at no time be used or occupied for manufacturing purposes, nor by a colored person or persons for any purpose. We will see ever used, never owned or occupied by persons of African descent, known as colored people, and by persons of African or Jewish descent, nor aliens accepting such as are descendants of the English-speaking race. We all know race is constructed. There is no English-speaking race for sure. There is one area that we found near the Country Club golf course, sort of northwest of Ann Arbor, that talks about 3/4 Aryan stock. So there are thousands of these in our county. There are thousands of them across the country, and they're part of a network of projects that is trying to understand these. Now, these were old. These were made unenforceable by a Supreme Court case in 1947. They were made illegal by the Fair Housing Act. So why do these matter? Well, think about the access to wealth that homeownership provides in this country. And if large portions of our community, primarily black folks, other people of color, and Jews, couldn't access that wealth and how that wealth builds over time, we think about how much is based on housing value in this community. We've also been part of projects that are looking at things like urban heat island effects and things like redlining and racist covenants. And the temperatures in covenanted areas are on average much lower than places that were not covenanted. And so we see all these ramifications. But more sort of philosophically, perhaps, it's also what kind of communities do we want to see, and how do we improve our communities and reckon with the past as part of this project? So that's really what we're trying to do with this. So you're going to see a lot of examples of information shared about this. But what we're really trying to do is help our communities build a better community for everyone here in Washtenaw County in a way that reckons honestly with the past, the sort of structural past that we see in these documents and other kind of housing practices that created the segregation we see today. So as I mentioned, back in 2023, we're like, there's this great project coming, and you're going to help us transcribe these deeds. And so that was that. It ran from February 24 to April of 25. And so we put in around 12,000 documents that we suspected had a racist covenant in them. And people went through and they said, there's a covenant here. This is the geographic address, type and address of it. These are the buyers and sellers because we want receipts. This is the date. And then the actual covenant text, because we can see how things change in different areas, how it evolved over time. Where there was case law, actually seeing the language change because of that case law. And so a few different results that we can share with this. So we partnered with the Washtenaw County Clerk's Office, shout out to Larry Kestenbaum and his staff, for providing us a hard drive of over 650,000 digitized property records, which we then ran through a whole bunch of complicated technology stacks to get around 12,000 that we suspected had a racist covenant in them. And then more than 1,000 of volunteers, just like you, did anybody else help transcribe these? Someone raised their hand in the back. And so we had a number of community transcription and documentation sessions where we got together with folks to do this in community and to be able to reckon with this and actually see this literally in black and white. And see that this, what people were really shocked by, that this was in one of our most intimate spaces, our homes, that we would see this kind of language. And it was this incredible encapsulation of the sort of systemic racism we talked about, as well as the individual racism, and both of which have created such divides and injustices in our country and in our communities and states and country. Each one of these documents had to be transcribed 5 different times. So we ended up needing around 62,000 classifications. And then in total, volunteers contributed more than 5,000 hours to making this project possible. This is a quick graph of where the covenants were over time. And we see right about here, I'm going to stay in the line, right about there, we start to see this pick up. That's 1920s. In 1917, there was a Supreme Court case that said racial zoning was illegal. And so you couldn't do this. The government couldn't say, Only black folks can live here, only white folks can live here. So it went to the private market. And so that's why we see the covenants explode in the 1920s. They drop off because of the Great Depression, and they rise again in the 30s and in the 40s. And then you also see like 47 is sort of above the 12,500, but they weren't rendered illegal until the 60s. So we actually see these being put on afterwards. And because people are like, well, make me take it off. So we see that here in Ann Arbor. Eberwhite neighborhood has one of those. We have a whole lot of stories and histories in these documents. Lots and lots of well-known names were buying and selling these properties. Gallup, Reinerson. Reinerson is on the name of the stadium in EMU. Zwerdling, Almendinger. Almendinger was one of the worst. We'll see on the map in a second. Dolph Nature Area. Kelkins, which owns a pharmacy next to the clothing place on State Street. Why am I blanking on the name? Van Boven. He's in all of these. Schlanderer, Morton, Tuolumne, Granger, lots of places that have streets named after them. Darling of Darlington. Lather is a street in Ypsi. All of these names are in these documents. We also have these sort of really incredible stories like Harold Wright and his family, who lived in a house in the Ainsworth Park area, just sort of southeast of downtown Ypsi. A group of white homeowners in the neighborhood sued to get him and his family thrown out of their house. And a law student named Carol Guarneri did some really amazing research and found out about much more about this, and they actually never left. Like, we have the court document that said you need to leave, but they never did. And so we know that there was more research to do, and the local chapters of the NAACP were publishing multiple articles on this. There were lots and lots of court cases and things around this. The Frank Lloyd Wright House in Ann Arbor, the Palmer House, that has a covenant on it. Public schools have covenants. Huron, Pioneer, Carpenter, and a couple others. The interesting thing about Huron is that we found that out because they actually repealed the covenant on some land that became Huron. And lots of University of Michigan involvement. When you see the map, there will be a big covenant right around the football stadium because that is on land that they bought that was covenanted. They knocked down the houses, but that land was still covenanted. They also purchased a foreclosed subdivision in the 1930s and had a long debate between regents whether to keep the covenants, and they ended up saying they had a moral justification to keep them because of the contracts that were signed, not really worried about the covenants of the racist part. We also have like Beale and Bonesteel in these documents and things like that. People who are former regents, roads named after them, things like that. And there are so many more. So, a handful of publications and tools that you all can use to learn more about this and perhaps take action yourself. This is kind of hard to see, but anytime you see something orange, that is where a covenant is. This is Ann Arbor and Ypsilanti. Stadium is there. That's the Dolph Nature Area. We see a lot around West Willow, a lot in Ypsi, a lot in sort of College Heights down there, EMU area. So all of these, are things that where there is a documented covenant. We also have them throughout the county as well, but you will see the most of them are concentrated around Ann Arbor and Ypsi. And thanks to our friends at the Ann Arbor District Library, shout out to Andrew and the archives group and the developers he works with to create this. We have an archive of every single covenanted document and property record on the AADL. This one is between Mabel W. Holmes, also known as Mabel White Holmes. Anybody know who Mabel Holmes is? Yep. She is, we see a lot of her names on covenants out in Chelsea, and she is the inventor of Jiffy Mix. She is the inventor of Jiffy Mix out in Chelsea. So you can go look up, so there is a link. When you click on one of these properties that are filled in, you can see a link to go look at the original property record from this. We also have a public data set of every property that we mapped. And so it's on GitHub right now. You can go download it. This is with our friends Mapping Prejudice in Minneapolis, who have been doing this work for almost a decade now and work with counties like ours and around the country. We also have a repeal toolkit. So if you have one of these on your property, you could remove it. Now, when we say remove, we mean add another piece of paper into your chain, title chain, things like that, to say, we renounce this. What we don't want to do is get rid of it. There are other projects that like, we're going to actually take all of this out and take this off the historical record. We hate it. It's like, yes, we hate it too, but we want it to still be there. And actually, the way this project got started, the very first ones we found, we partnered with Liberty Title, and so we were looking through some of their records, and we found them by either because Liberty Title or someone else had literally tried to X them out and redact them, but they didn't do a good job, and so we could then figure out what it said. So this is a toolkit that takes advantage of a recent law passed to make these repeals and discharges easier. We have this whole thing on our website, including some forms to fill out. You can do this on your own, or if you live in a neighborhood that might be fully covenanted, meaning that every property has a covenant on it, you could work with your neighbors to repeal that as well. So this is the suggested language. We disavow the language and replace it with this. So this is the Hannah subdivision repealed their racist covenant 3 or 4 years ago. So this is one way you could do this. But we also know, oh, here you go. I can go back if you want to take a picture of that. Okay, cool. We also have community conversation guides, because repealing, can feel like an important step, and it is, but that often benefits the people who benefited from these practices and not the people who were victims of these. And so we often say, and I'll give a shout out to my colleague, Jess Letaw, who talked about this, where, okay, I found my house on this map. What do I do? And then the other option is, well, I didn't find my house on this map. What do I do? And so we have community conversation guides on the Justice Indeed website where you can talk to your neighborhood group, your church, your school, other forms of community gatherings, to talk about what this history means for you, what it means for your neighbors, what it means for our communities. And what's next? We're kind of figuring that out, and we need you to help us do that, because we have all of these things that we have made available to our communities and to around the country. I just had a conversation with someone who are working on, again, the climate-based harms that were done by racist housing practices. It's like, can we get access to the data sets? Like, yep, here you go. See what you can do. And there are people who want to do more research on these different parts. And so we would love to hear your ideas, both whether it's tonight or in general, about where you would want to see this kind of work go and how we can make our communities better. So thanks very much, and I'd love to hear what you have to say. Sure. I'm told 2 or 3 questions. Yes. Can you say that one more time? So the question was, why would non-covenanted areas be hotter? That's because there's more concrete. So covenanted areas have things like parks and green space and things like that. So you also see this with like redlined areas that have less of the quote-unquote amenities like parks. So less things that block the heat out. Other questions? Yes. Yes. Mm-hmm. Because that is removing it from the historical record, which we don't want to do. We want it to be part of the historical record as a record of these practices. That's why. He asked if why not just simplify things and remove the covenant from the record. And I said the point of that is to keep it on the historical record, because we don't want to remove it as a way of whitewashing the past. So I see two hands. I will take them both, and I'll try to answer them both. So in the pink first. Sure, I will do that. Go ahead. Mm-hmm. Yes. So first redlining. Those were literally federal lending maps that drew lines and said these neighborhoods we will actually insure your mortgage, and these ones those were colored green. And then when you got to the red, literally red line neighborhoods, these are the ones they wouldn't insure. So we can see those around the country. We don't have them in Ann Arbor. We weren't big enough. There's some in Detroit, Jackson, Lansing, and I think one more city. There are redlining maps. We do not have them in Ann Arbor. And the other question was around workaround language and seeing about other ways that that language could be used to restrict folks. Our method was really looking at like 40 to 50 kinds of phrases that we found when we OCR'd all of these documents. And so if it doesn't have some of those phrases, we're not going to find it. I would love to talk to you more about that and what kind of examples you had in mind, because we are also looking at, because we still have all this OCR text, and people are like, let's see what the banks are involved in this, and like see these other kinds of institutions that might be part of this, or are looking at other forms of housing history in our communities and what else we could learn from this. So I would love to talk to you more wherever you went about, okay, I see you, I'm gonna come find you. So, oh, I see the person back there. So, do we have time for, okay. So real quick, the question was around corpus linguistic tools. I'm going to answer this slightly differently, which is that everyone asks, can AI do this? And the answer is sort of, but not really well. There was a big paper that was like, we can do all this stuff, we don't need people. And it's like, oh yeah, actually you don't get down to the level of detail that human volunteers do. So the answer is no. We are looking at, what I'm interested in with that kind of work is seeing variation across communities. And so what we are looking at when we have all of those data sets online is seeing the differences. So just one example there is, and I'm going to be real quick, there was a developer in Tucson that came from California and imported the language from California, including like oil wells and things like that, which there aren't oil wells in Tucson. At that time, there weren't anyways. And so seeing where this kind of geography of exclusion and racism sort of can be seen in these documents. So happy to talk more. I've got stickers for Justice Indeed and other kinds of things. Thanks, y'all.
All right. And we are up to 8 o'clock. We've got 10 minutes, so please refresh your drinks, meet a new person that's sitting around you, engage in an interesting conversation, and we will see you back here at 8:10. Again, make sure you say hello to our wait staff, tip your servers, and we'll see you in 10 minutes. get back after this break. And before we get our next speaker up here, I want you to all do me a favor. Take a deep breath in and hold it. Bask in the glow of cellular respiration. ATP synthesis. Isn't it great, folks? One of life's necessary processes. Exhale. And now you all have a deep and personal connection to our next topic. Oxygen. Is it good for you? And without further ado, we're going to welcome from the University of Michigan a microbiologist and science writer, Ananya Sen. Ananya, come on down.
All right. Hi, everyone. Yes, I am a PR representative at Michigan Medicine, but today I am going to condense what I learned for 7.5 years doing a Ph.D. in microbiology in about 15 to 20 minutes. So let's see how that works out. And I'm also going to bookend the very first talk. We're going to talk about microbes yet again. So most of us in high school, probably, learned that oxygen is used by our cells as a source of fuel. So oxygen reacts with glucose to release energy along with carbon dioxide and water. Now, there's a problem with using oxygen to do this. Yes, you get energy, which is great, but during that process, you also generate reactive oxygen species. And I have two examples. The first one is hydrogen peroxide as well as hydroxyl radicals. So why are they dangerous? Why are reactive oxygen species dangerous? I'm going to start with hydrogen peroxide. So this cell at the bottom is a bacterial cell, just a representation. But the thing to remember is whatever is happening in a bacterial cell is also happening in all of your cells. So all of our cells have a large amount of iron 2 inside of them. Now, this iron 2 is found on the surface of the DNA in this bacterial cell. It's also present in a lot of different enzymes, and these are called iron-dependent enzymes because iron is a great catalyst. Why am I telling you this? It's because iron can react with hydrogen peroxide to form iron 3, as well as hydroxyl radicals. And hydroxyl radicals are extremely reactive. They can react with anything they touch inside the cell. And that's why reactive oxygen species are dangerous. And you have several examples of oxidative stress in your environment. So if you go down CVS, you're going to see bottles and bottles of hydrogen peroxide. If you ever get a wound, you pour some hydrogen peroxide on top of it in order to disinfect it, to get rid of the bacteria. But if you notice, sometimes you'll see bubbles that come up. And those bubbles are because you have catalase inside your own cells that's basically getting rid of the hydrogen peroxide stress. This is also true when you go get an x-ray. You are getting an x-ray for about 5 minutes. The x-ray technician is standing in a different room because they're not supposed to be there, and this is why. X-rays can basically act on the water inside of their cells, breaking it into hydroxyl radicals as well as hydrogen peroxide. This is an example of the kinds of experiments that used to be done with oxidative stress. This is from 1947. The two scientists are trying to figure out how much oxygen can you give a deep sea diver. Completely unethical. You shouldn't be doing this. But the man in the middle is being given 100% oxygen. The results of this are pretty interesting. The first thing they notice is lung toxicity. The man had problems breathing, he had some coughing. He also had behavioral problems, suggesting that there was nerve toxicity because of the amount of oxygen. Oxygen therapy, something that still exists today in the medical community. You use it in anesthetics. So, for example, when a patient is under anesthesia, sometimes their blood oxygen level will dip down and you have to give them supplemental oxygen. Premature babies, the very last thing to develop in a baby is the surfactants on their lungs, so you have to give them supplemental oxygen. The problem is, in the past, overshooting how much oxygen you give patients used to be a problem. And now, as we've understood what oxygen therapy needs to be doing, how much oxygen to give, it's not a problem anymore. But all of you do know a famous example of this. Stevie Wonder, as a baby, was given a little too much oxygen and it damaged his retinal cells, which is why he's blind. Now, your bodies know this very well. They know how dangerous oxidative stress is, which is why when you have pathogens, bacteria, that come into your body has white blood cells that will just literally hose them down with hydrogen peroxide because your body understands that it is very dangerous. So now you can probably be wondering, well, it seems like the problem is that you have all this iron inside the cells. That's kind of dumb. Why did cells do this? Why did our bodies do this? Why did bacteria do this? Well, in order to answer that question, we have to walk back in time to about 4 billion years ago and look at how life evolved. So life evolved in a world that was completely devoid of oxygen. You had a lot of carbon dioxide, you had methane, you had hydrogen, you had sulfur from all the volcanic eruptions, and you also had a lot of free iron too that was present in the oceans. And bacteria looked at this iron too and said, you're pretty good at catalyzing reactions. We like that. We're going to use you. We're going to use you in our cell processes. We're going to use you in our enzymes. Not a problem. However, 1 billion years later, I would argue, was the first example of climate change. Some bacteria figured out, hey, we have sunlight, we have water, we have carbon dioxide, we can do photosynthesis. And the byproduct of photosynthesis is oxygen. So oxygen started getting released very slowly into the atmosphere. The first consequence of that is that oxygen reacted with all the free iron 2 that was present in the oceans, converting it into iron 3 that precipitated out. But the other consequence is now oxygen is attacking all these iron-dependent things inside the bacteria. But they've been doing it for 1 billion years. They're not going to change that. That's really hard to do. So the bacteria figured, we'll deal with it. We'll figure something out, but we're not letting go of our toxic iron dependency. So 1 billion years later, you have basically the atmosphere we have now. You have 20% oxygen. Oxygen is still a problem. It's basically reacted with all the iron that's present free in the oceans. It's all precipitated out as iron 3, and bacteria have figured out what to do with all this oxygen toxicity, and that's basically what our cells have also learned how to do. Now, you can see these iron precipitates. If you go to the Utah-Arizona border, you're going to see these gorgeous red cliffs, and they've been formed by all the iron 3 that precipitated out billions of years ago. So that's why hydrogen peroxide is dangerous, because we have this dependency on iron, because iron is a great catalyst, and so our cells have figured out ways to deal with this stress. So our cells actually are very good at having antioxidant activity inside of them. So in order to talk about what that antioxidant activity means, how do we know hydrogen peroxide or hydroxyl radicals are a problem, I'm going to talk to you first about how scientists figure all of this out. Rule number 1, we don't experiment on humans. It's a terrible idea. There are better things to do. You use model organisms. The reason we use them is they're much easier to work with, it's not unethical, and they grow pretty quickly, so you get your results a lot faster. Examples of this include E. coli, which is what I worked with in my Ph.D., as well as fruit flies. Now, you may be wondering, why? Why do we use these model organisms? Well, a very famous scientist called Monod once said, what is true for E. coli is true for an elephant. And that means all the cell processes that occur inside bacteria, more or less, with a couple of minor tweaks, happen inside our cells as well. So if you study it in bacteria, you're going to have a pretty good idea of what happens inside your cells. So going back to what classes of damage are seen, in order to understand the first class, we're going to have to look at cell division. So in a normal bacterial cell, during cell division, what happens is the cell will make 2 copies of the DNA, and then it'll split in half, making 2 daughter cells. If there's DNA damage, the cell's like, hold on. We are not going to make 2 copies of this damaged DNA. We're going to spend time repairing this DNA, and so the DNA gets repaired over time. The rest of the cell doesn't get that message, so it keeps becoming longer and longer in a process that's called filamentation. And you can see this under a microscope. So these are E. coli cells in the absence of hydrogen peroxide, very short rods. If you add hydrogen peroxide, they form these beautiful long filaments. You also have enzyme damage. I'm not going to tell you or bore you with the names of these enzymes. They're 2 different classes of enzymes. But I do want you to recognize that in the first case, if you add hydrogen peroxide, the activity goes down from 100% to 18%. And for the other enzyme, the activity goes down over time of exposure of hydrogen peroxide. So just 5 minutes of exposure can knock down the activity by 60%, and that activity keeps decreasing the longer the enzyme is exposed to hydrogen peroxide. So how do our cells deal with it? How do bacterial cells deal with it? There are 2 major ways. One is you remove the stress, or you repair the damage. The stress is being caused by the reaction between iron 2 and hydrogen peroxide. So if you want to remove the stress, get rid of one or both of these. And E. coli, fruit flies, and humans all have ways of doing that. We have enzymes called catalases and peroxidases that can get rid of the hydrogen peroxide. And we have entire proteins that will basically just hold on to the iron and protect it inside a shell so that it does not come in contact if hydrogen peroxide is around. The other way you can do it is you deal with the damage. So there are 2 classes, major classes of damage. You have DNA and enzyme damage, and our cells have enzymes that can repair the DNA, that can repair the damaged enzymes, and this is true across the board in different organisms. So you may be wondering, what do scientists still study oxidative stress? Why do they do that? Well, the reason is that most organisms have figured out defenses that are just about enough to deal with the oxidative stress that they encounter on a daily basis. So if you think back to that experiment, humans know how to deal with 20% oxygen. It becomes a problem when they have to deal with 100% oxygen. That's also true of all the pathogens that are constantly attacking us. They kind of know how to deal with the oxidative stress that's present in their environment. However, when they come into our bodies and get sprayed down by hydrogen peroxide, most of them die. Most, but not all. Salmonella and Mycobacterium tuberculosis can actually survive inside our body. They're pretty resistant to all the oxidative stress our body throws at them. And so the question is, well, why? Why are they so resistant? What can we do? And that's why scientists are still trying to figure out what are the defenses against oxidative stress, especially in these pathogens. However, if you ask Wikipedia, hey, what different diseases are caused by oxidative stress? Basically says, yes, all of them. Now, these are all conjectures. There's not a lot of great proof that oxidative stress is causing this. And the reason is that it's really hard to study oxidative stress. There are not a lot of great techniques. There are not a lot of great dyes. And it's very hard to show that, yes, oxidative stress is the only thing that is causing damage in any one of these. And so that's why scientists still have to actively study this and try to figure out whether oxidative stress is involved in any of these. However, that has benefited one particular industry, which is the antioxidant industry. You have antioxidants in your tea, your coffee, your skincare, antioxidant water, chocolate, anything and everything. And really, it doesn't make any sense if you think about it, because we've evolved for billions of years to figure out how to deal with oxidative stress. If you haven't noticed, you're still all alive. So your cells know how to deal with the oxidative stress that they're encountering on a daily basis. Taking in an additional source of antioxidant is not going to help you. I mean, the term, you could just put it on anything and make any claim. You could put it on your pets. I would argue there are more studies that tell you that your pets are better for your health as opposed to antioxidants. So basically, if you want to have the antioxidant chocolate, if you want to drink the antioxidant tea, go ahead, do it. But do it because you like doing it. Don't do it because it has antioxidants inside of them. And with that, I have to end with my favorite oxidative stress meme, and thank you all for listening.